Okay, so it is 7 p.m. So I think I'm going to start with the introduction. Hello, everybody. My name is Alexander Haber. I'm a postdoc from the Washington University in St. Louis. And together with my friend and colleague, Andres Malagon, we are organizing Astronomy on Tap in St. Louis. Today, we have two very interesting talks. The first one is going to be by Johanna Nagy. She's our newest faculty member at the physics department of Washington University in St. Louis. So she's going to tell us something about the oldest light in the universe, the cosmic microwave background. And the second talk we have is by Daniel Agustinho, who is a postdoc at the Med School of Washington University in St. Louis, who is going to talk about space fungi. How can something grow where it's impossible to survive? If you have questions, please just post them in the YouTube chat. We will monitor the chat and we will forward your questions either during the talk if it's really urgent or necessary, or otherwise at the end of the talk, and our speakers will gladly answer your questions. I will take just a short time to say thank you to all our partners and sponsors. First of all, Washington University, then Urban Chestnut Brewing Company, where we normally are holding these events. Right now, unfortunately, due to the pandemic, you're all responsible for your own beer. So cheers to everybody. Normally, there are also other events happening in St. Louis, especially one of our partners is Science Distilled, but I think they're on a break during the pandemic. Otherwise, we want to thank the Solar System Ambassadors, the Academy of Science for St. Louis, and our newest partner, the McDonald Center for the Space Sciences. Okay, so I think we are ready to start with our first talk. So our first speaker is going to be, as I said, Johanna Nagy. And Johanna, please start sharing your slides.
Great, so hopefully that looks good. So tonight, I'm really excited to tell you about the oldest light in the universe. And so first, I'm going to tell you a little bit about this light and where it comes from. And then I'll tell you about the instruments that we use to study it. And for those of you who are curious about my video background, it's a picture of the inside of my lab. And it's where we build telescopes like this to study the earliest light. So since we can't all be together tonight at the Urban Chestnut, I figured that this would be the next best place. So, okay, so my talk today is all about cosmology. And I'm a cosmologist by training. So that means that I study the universe on the very largest scales. So when you think about outer space, you might think about objects like planets or stars or maybe galaxies. And I try to understand the even bigger picture to answer questions about what the universe is made of and how it's changed over time. So most of what we've learned about the universe so far comes from looking at light. And we're fortunate that we're not just limited to light that we can see with our eyes. There are other colors of light that we can see with specialized telescopes. And so we can learn a lot about the universe by combining information from all across the spectrum. So the light that I'm going to focus on today is right here. It's in the microwave part of the spectrum. And as the name suggests, this is similar to the light that your microwave oven uses to cook food quickly. And it's also similar to the Wi-Fi signals that might be letting you watch this talk today. So humans are pretty good at producing microwave light. And it turns out, so is the universe. So when we look up at the sky with our microwave telescopes, we can see a signal in every direction. And it's not coming from any specific object because it's the glow of the universe itself. So this is very different from what we see when we look up at the night sky with our eyes. And since we don't have microwave eyes, I had to pick a color that we could see for this picture. So I picked green, but that was an arbitrary choice. So studying this microwave light has allowed us to piece together the story of our universe. And this is a story that's been unfolding for almost 14 billion years. So if you thought that 2020 was long, now just imagine 14 billion 2020s. But actually, and maybe luckily, the early universe was very different than 2020. So if we go back to when the universe was just a few 100,000 years old, there were no pandemics because there were no viruses, and there were no molecules or even atoms. The universe was so hot and so dense that all of the particles were in a plasma. And if you're trying to picture this inside your head, just imagine what it must be like just inside the surface of the sun. So this is what it used to be like everywhere in the universe. So in this diagram, you see a plasma, and it's filled with protons and electrons, as well as photons, which are just particles of light. And so these particles were constantly running into each other. So they're just all continually bouncing around. But as the universe got older, it was expanding, and it was also cooling. So eventually, it became cold enough for the protons and electrons to stay bound together in hydrogen. This hydrogen gas was transparent to the photons, so they stopped bouncing around. And it was as if these photons suddenly just got really good at social distancing. So in fact, they got so good at it that once hydrogen formed, most of them spent their next 13 billion years without running into a single other particle. So compared to that, 2020 might not seem quite so long anymore. But for cosmologists, this extreme photon social distancing is actually really great. And that's because it allows us to use these photons to get a baby picture of the universe. So today on Earth, we see these photons as microwave light. And so we call it the cosmic microwave background, or the CMB for short. And when we look out into space from Earth, we see the photons that have been traveling towards us. And we can trace them all the way back to where they were emitted, which is at the surface of that dense plasma more than 13 billion years ago. So these are the oldest photons that we can see in the universe today. And when we look at them, we see the inside surface of the plasmasphere as it was the last time the CMB photons scattered. Now, from this picture, it might seem like the Earth must be at the center of the universe because we seem to be at the center of this sphere. But actually, if there were aliens on another planet in a galaxy far, far away, they would see a similar picture with them at the center. And that's because to any observer, they're at the center of all of the light they can see, which is not the same thing as being at the center of the universe. So anyway, we can think of the CMB photons coming from this plasma sphere as having a certain temperature. And when they were emitted, they were roughly a few thousand degrees. But today we see them as very cold. So the CMB that we measure is only about 2.7 Kelvin, which is negative 454 degrees Fahrenheit. So if we go back to this picture, that's the glow that I chose to color green in the picture of the microwave sky. But if we blow up the color scale on this picture, we can see it turns out that the CMB isn't exactly the same temperature everywhere. And we've actually been able to measure these tiny temperature variations. So you can see them in this image that was taken by the Planck satellite, was released in 2015. And this is a map of the microwave sky. It's our baby picture of the universe. So you might think it looks cute. But these colored splotches may look like noise, but they actually represent real temperature variations, which are pretty tiny. They're less than 1 part in 1,000. But actually, we expected these small differences to be there because we know that our universe isn't exactly the same everywhere. They're clumps of matter that are organized into structures like galaxy clusters. And the temperature differences in this picture show us the seeds from which the cosmic structures evolved. So by looking at the microwave sky, we've been able to piece together a lot about the history and the composition of the universe. And that's because the light from the CMB experiences most of the history of the universe as it travels to our instruments. So as you can see from this timeline, this includes when the first stars formed, later followed by galaxies, and all the way to the present day. And this might sound really complicated, but it's now been more than 50 years since the CMB was first discovered. So we've had a lot of time to piece this together and to learn how to make better and better measurements of it. So after 50 years of studying the CMB, you might wonder what's left to measure and why cosmologists like me are still obsessing over this ancient light. So for the rest of my talk, I'm going to focus on what we don't know and how we're going to figure it out. And the truth is that there's still so much that we don't know that I don't have time to tell you about all of it. So I just picked one thing. And that thing is called inflation. And it's a theory that could explain what happened when the universe was much less than 1 second old. So cosmic inflation is an idea that's been around for several decades. And in this theory, the universe would have undergone a period of extremely rapid expansion when it was less than 1 second old. It would have grown by a factor of more than 10 to the 20 times its original size. So my illustration here is woefully far from showing the scale of this expansion, because 10 to the 20 is this number. So that's really hard to illustrate on the slide or even to picture in your head. And to make this whole process even more incredible, it would have all taken place when the universe was much less than 1 second old. So we don't know exactly how old the universe was when this would have happened, but something like 10 to the minus 30 seconds would be a reasonable guess. And that's an incredibly short amount of time. That's this number. And so it wouldn't even come close to registering on a stopwatch. And all of these numbers are pretty incredible. But to me, the even more incredible thing about inflation is that we might be able to detect it with our telescopes. So to see how we can look that far back in time, we can return to this picture of the history of the universe. Remember that the CMB is the oldest light that we can see today, and it was emitted when the universe was a few 100,000 years old. But the epoch of inflation that we want to study happened when the universe was much less than 1 second old, so we can't see any light from that time. However, one of the amazing things about inflation is that it would generate a background of gravitational waves. And these would ripple through space and leave a special signal in the CMB when it's later emitted. So the key to this special signal is in the polarization of the photons. Photons all across the electromagnetic spectrum have a property called polarization. And you might be familiar with this if you have a pair of polarized sunglasses. These have a special filter that blocks light with a certain polarization to make it darker, but they still let through other light so that you can see. And it turns out that the CMB is also polarized, and so we can build special telescopes to look for this polarization signal from inflation. So here's a picture showing a map of the polarized microwave sky, which also comes from the Planck satellite. Planck has measured polarization signals in the CMB that are thousands of times smaller than the temperature variations that I showed you earlier in this talk. But despite this incredible sensitivity, there's unfortunately still no sign of a signal from inflation. However, even not seeing a signal actually helps us to learn about the universe. That's because it can rule out certain models of inflation that would otherwise have been possible. And just because we haven't seen a signal yet doesn't mean that it doesn't exist. That signal from inflation could just be too small to show up in these maps. So that's why we're still looking. And the reason that it's so hard to find the signal is not just because it's so faint. It's also because we have to be able to tell it apart from other similar signals. So for instance, this colored band here in the center of the map is not the CMB, but it's our own Milky Way galaxy. And it turns out that living in a galaxy is actually a huge problem for cosmologists. So when we observe the sky, we do so from Earth, or very close to it if we have a satellite. But our own Earth is embedded in our solar system, which is in turn embedded in the Milky Way galaxy. So if you go far away from the city lights and you look up at a clear sky, you might be lucky enough to see the dust and the high density of stars that appear when you look towards the center of our galaxy. And it's beautiful, but to cosmologists, it's also annoying. And that's because even though we live in one of the spiral arms of our galaxy, we still have to look through our galaxy at least a little bit, no matter which direction we look. Our telescopes have gotten so sensitive that we can see the signal in front of the CMB signals that we're hoping to measure. So separating these so-called galactic foregrounds from the cosmological signals is a major challenge for modern experiments, but we've developed some techniques to do it. So speaking of modern experiments, for the rest of my talk, I'm going to show you some pictures of instruments that we hope we might use to measure this signal from inflation. And this is such a big puzzle in cosmology that there are actually many different instruments trying to do it. and they all work in slightly different ways, but I'm just going to tell you about 2 of them today. So this is an instrument called SPIDER, which flies on a balloon above Antarctica. And to give you a sense of the scale of the instrument, you can see a person in this lower left corner. And as you look toward the center of the instrument, what you're seeing is 6 telescopes that can measure CMB polarization. So each telescope has an array of hundreds of detectors that are actually a few degrees colder than the CMB itself. And each of these telescopes has 2 lenses to focus the light onto the detectors. And then these telescopes are supported by a lightweight carbon fiber structure that controls where they point on the sky as they're hanging from the balloon. So this picture shows SPIDER just before it launched for its first flight in 2015. And we're hoping to fly it again when this pandemic is all over. So for those of you who are wondering about my title slide, this is the same picture. And this is showing you SPIDER's view from the stratosphere, which was taken by a GoPro camera during the 2015 flight. So this picture shows what you would see with your eyes if you could fly with SPIDER. So it's made with optical light. This isn't a microwave picture. But it's also possible to measure the CMB from the ground. And even though you have to worry about the atmosphere when you're on the ground, you can build bigger instruments than you can put on a balloon or on a satellite. So we're also starting to build a new collection of ground-based telescopes called CMBS-4. And we hope they'll be able to start observing the sky around the middle of this decade. So there will be 21 telescopes with several different designs, and they'll observe from both the Atacama Desert in Chile and from the South Pole. And both of these sites are high and dry, so they're some of the best observing sites on Earth for microwave telescopes. And together, all of these telescopes will deploy hundreds of thousands of detectors. And so will be sensitive to signals from inflation that are more than 100 times smaller than what we can see right now. And there are, of course, a lot of technical challenges with a project of this scale, but there are hundreds of scientists from all over the world who are working together to make this new project a reality. So since I want to have plenty of time to answer your questions, I'm going to end here with a quick summary of what we've just talked about. So the oldest light that we can see today is the cosmic microwave background. And it gives us a picture of the universe when it was only a few 100,000 years old. By studying this light, we've been able to learn about the structure and composition of the universe, but it still has a lot to teach us. So by looking at the polarization of the light, we can search for a signal from inflation, which would have occurred when the universe was much less than 1 second old. And the instruments that we're building today let us trace these signals back through cosmic time to piece together the story of our universe. So thanks so much for listening to my talk tonight, and please post your questions in the chat. Do I have any questions so far?
Okay, thanks very much. That was a really fascinating talk. So since there's a short delay, I'm gonna abuse my status as an MC to ask the first question. So you said the telescope has to be colder than the CMB itself, is this right?
The detectors are colder than the CMB, that's right.
So, I mean, this, as you said, the CMB is really, really cold, right? So even where the balloon flies, I guess it's not cold enough to reach that. So how do you cool the instruments on the balloon to achieve these low temperatures?
Oh, that's a great question. So I'm going to go back here actually to my picture of SPIDER. So part of the reason that this segment in the middle that houses the 6 telescopes is so big is because that has to actually cool the detectors. So that is a giant container of liquid helium, which is actually, it's 4 Kelvin. So liquid helium is a little bit warmer than the CMB, but then we have special refrigerators that start at 4 Kelvin, but then they get much colder. So they go all the way down to about 250 millikelvin. So that's just barely above absolute zero. And that's where we operate our detectors. And so it's these special refrigerators that are housed inside that that big container that actually let us cool our detectors.
This is still above the superfluidity transition of helium?
So it is not, it is below. And we actually have a special tank of superfluid helium that we feed from our normal tank.
So this does not leak out and it's okay to contain. Superfluid helium is normally rather tricky to keep.
It is, you have to be very careful when you're making this container, but it can be contained.
Oh, great. Okay, so there are the first questions in the chat. So the first question is, how big are the telescopes that you're using? I think this has partially been answered by these slides. And how long does a typical measurement take?
Okay, that's a great question. So yeah, for the scale here, you can see the person in that corner. So this is a balloon-borne telescope. So in terms of the weight of this telescope, we're limited to only about 6,000 pounds. So it might sound like a lot, but it's really not, because some of the telescopes that we put on the ground are actually much bigger. So the bigger we can make the mirror, our lenses, the primary optic on our telescope, the better resolution we can get. So on the ground, we can have telescopes that are much bigger, up to say 10 meters in diameter. So then the second question was how long for these measurements? And that depends a little bit on the instrument. For a balloon, right now, the longest we can stay up is approximately a few weeks. But if we could stay up longer and keep our detectors cold for that long, we would take more time, because the more time we get, the lower and lower our noise is. So the more sensitive our instrument can be. On the ground, these telescopes would observe for years. And so it's basically, they stare at the sky and they can integrate down that noise to do a very sensitive measurement.
And we also have a follow-up question to that. And how long does data processing take in contrast to the actual measurement?
Right. So data processing can also take a long time. It can potentially take years to process all the data. Part of that is because we have so many detectors, so it can be a lot of data and we're not looking through it all by eye. We have computer algorithms that help us. And part of it is that we need to do a lot of checks on our data to make sure if we're seeing signals that they're really coming from the sky. And if we see anything, we would want to know that it's really coming from inflation rather than our own galaxy or our instrument. So a lot of the time is just spent checking the data and trying to subject it to all of the tests we can to make sure we're really confident in our results.
So we have a question from Jeffrey Wright. Would LIGO or a similar facility ever be able to detect the gravitational waves from inflation?
Okay, so LIGO has seen some really cool gravitational waves from merging black holes and neutron stars. And so these all happen in the sort of the much more recent universe, we'll say, than the inflationary universe. And they're also, these events are much higher energy. So the gravitational waves from inflation would be very, very hard for an instrument like LIGO to detect. So right now it's way beyond the sensitivity of LIGO. But if the question is, could we ever see it? Well, in principle, eventually, maybe yes, but certainly the technology we have is a long way from that.
So would LISA, the space-based gravitational wave detector, be able to see these kind of waves, or is this still not sensitive?
No, it's still, these are much lower in frequency as well. So it's kind of well beyond the reach of those instruments. But in a way, it might be good for their science, actually, because they're looking for very different events, whereas these gravitational waves are sort of a background. So they would never be able to make these go away.
We have a question from Sarah. You mentioned that you want to do another round of images after the pandemic is under control. How has COVID delayed your research?
Right. So we were hoping to fly Spider this year from Antarctica, but there are no balloon flights happening this year because of COVID. So unfortunately, it has delayed us. But as soon as the pandemic's over, then we're hoping to get the instrument in the air as soon as possible.
So are the bases in the Antarctica, are they empty right now, or are they still crewed in any way?
There are skeleton crews down there. There's sort of a minimum number of people down there needed to just maintain the station, the facilities. But yeah, not a lot of scientific operations are happening this season.
We have another question about the detection of these waves. So can the pulsar timing array maybe detect these massive gravitational waves?
I don't think they'd be able to detect the ones from inflation. And so really the CMB is our best hope for seeing these particular gravitational waves. But again, it also depends. We don't actually know how big the signal is. We just know that we haven't seen it yet.
Do you remember correctly that it was already a couple of years ago a claim that these waves have been seen in the CMB, which had to be retracted then at the end?
That's right. So the BICEP2 experiment a few years ago had seen an excess signal in the CMB polarization. And at the time, they thought that it might be from the gravitational waves in the early universe, from inflation. But that was before the maps from the Planck satellite had been released. And so with the information from Planck, they were able to then say it was actually consistent with signals from our own galaxy.
So are you hoping for an even better resolution picture of the CMB in the future from other satellites as well? Or?
So certainly that would be wonderful. With satellites, they take a long time to actually get in the sky, even once they've been funded. So right now, there's no planned funded satellite experiment that would see this. People have ideas and there are things that are being talked about, but there's nothing for sure. And so what we can say is that another satellite, so kind of a follow-up to Planck, would then be many years down the road. But in the meantime, there's still a lot that we can do from balloons and from the ground.
So Fapeng Wang wants to know, are there any proved CMP projects to measure the CMP spectral distortion in future?
So that's another thing that people have been talking about. So instead of looking at spatial variations, you can look at the frequency of the cosmic microwave background. You can see how close it really is to a thermal black body. So again, that's another project that could be done really well from a satellite, but it's a very small signal. And so there are plans and people are proposing things, but there are no funded experiments right now. So there's nothing for sure that we can say is going to happen and when it's going to happen.
Great. I don't see any further questions right now.
Great.
So,
okay.
Thank you very much for this talk. It was super interesting.
Oh, thank you. And I will stop sharing my screen.
Thanks. In the meantime, we have a quick survey. If anybody wants to give us feedback and answer a couple of questions about these events, I'm gonna type this into the chat now. So just if you have time, please fill out the survey. And oh, we have one last question. So can you tell us a little about your background and how did you end up working in this research?
Sure. So I think the reason I ended up working in this research, I think, is because when I was an undergraduate student, I took a class called Hands-on Electronics and realized that I really loved to solder. And I thought it was just so cool that I could take something that looks like a pen and melt metal and put together these circuits. And so I thought I wanted to try research out during the summer. And I saw a research group that was advertising that they needed someone to help with soldering, to make some stuff and to do some other things. But it turned out that it was actually doing research. It was basically doing experiments that are very similar to what I'm doing now.
And how are these studies coordinated with other countries?
So in terms of like day-to-day coordination, even before the pandemic, we've been doing a lot of stuff virtually, precisely because we have so many people working in so many different time zones and on so many different continents. So even before the pandemic, we were having Zoom meetings, we were on Slack all the time. But then there's coordination at sort of higher levels too, between government funding agencies and basically people working to make sure all of these experiments happen logistically. So there's a lot of coordination, even in terms of shipping stuff around, in terms of figuring out what gets funded. So it's definitely a very collaborative effort.
Great, thank you so much again. I think it's time that we move on to our second topic, which will be a little bit different. Daniel Agustinho is going to talk about space fungi. So Daniel, please start sharing your screen and take it away.
All right. Thank you for the introduction, Alex. I hope you can hear me well. So I'm Daniel. I am a postdoc here at the Washington University in St. Louis at the Department of Microbiology, and I work with one of those space fungi that Alex was mentioning. And although I'm not looking at how they survive in space, it's something that has always been like a curiosity for me, like to study these aspects of this fungus that we work with. In my lab, the Durian lab, we actually study the fungus as a pathogen, not so much as a space explorer. So we're talking about space fungi and how something can grow where it's impossible to survive. Spoiler alert, it's not so impossible apparently. A lot of things can grow, a lot of organisms can grow in situations that are similar to space or very extreme situations that we can find on Earth or in other places in the solar system. And our story actually begins in Chernobyl. The year is 1991. That's exactly 5 years after the explosion that destroyed, let me point out here, that the explosion of uh, the the, the oh, of the facility of the nuclear plant, and 5 years after that, there was the first investigative team that went to the local and were able to investigate what actually happened. And it's amazing to think that like 5 years before, it was such a toxic environment. It's the radiation was so intense that a human being would have died in less than a minute of exposure. But 5 years later, it was actually possible to send someone there with the proper red with the proper equipment. This group that went there in 1991, they were kind of shocked to find out that the whole structure of the power plant, and especially near the core, there were a lot of fungi growing, specifically black fungi. They saw some mushrooms and mold growing and feeding on the graphite structure that surrounds the core of the reactor, and also the cooling water of said reactor was completely black, like darker than red wine, because of fungi growing in this structure. Uh, right. And so I'm talking about fungi, but I wanted to specify what are fungi. Uh, there are three different kinds, three biggest categories that we work with. The yeasts that here are represented by Saccharomyces cerevisiae, or also known as the baker's yeast. And well, it's famous worldwide for making beer and bread, so their importance cannot be uh, not taken into consideration. There's mold, as you can see here on the on the top right. This is actually a picture from the International Space Station where the black mold growing, where they normally put the towels after showering. And mushrooms that you can find here. This is a picture taken closer to Chernobyl. So all of those are fungi, and some of these fungi can actually change between these morphologies, mostly between yeasts and the mold structure, as a response to the environment as they need to. And they have specific structures that will help them survive and in conditions such as the high radiation in the center of Chernobyl. The surrounding area of the power plant, you can find lots of fungi that have this dark color. So here we see some mushrooms, but also the mold, as I mentioned. And they are black. I'm going to mention why. When they started studying those organisms that were growing near the power plant, they realized that they had a radiotropism. Tropism means growth, but in this sense, it's like they realized that these fungi, they were growing towards the radiation. So they had experiments in where they had some mold or some mushrooms, and they would have a source of light radiation similar to the one that was produced by the core of the power plant. And they realized that 66% of the strains of fungi that they were analyzing, so 18 out of 27, were actually growing towards and not running away, let's say, from the radiation. Think about plants, how they grow towards the sun. That's pretty much what they were observing. So they had this fungi growing towards the radiation source. They also observed something called radio stimulation. So fungi, they produce something called spores. They are like smaller cells that are pretty much inactive, like they don't have a metabolism. They're like seeds. It's a very nice metaphor, like seeds from a plant. So the spores from the fungi, they are used mostly for dissemination, for dispersion, and they are extremely resistant to environmental stresses, such in this case as radiation. And they observed that these spores were germinating like a seed germinates when it finds the ideal conditions for growth. They were germinating when there was radiation involved. So radiation was acting as a positive, again, not as a stress, but as a positive reinforcement from the environment for these cells to germinate. Later on, in the 2000s already, the first papers, the first researchers started coming, showing that there was something called radiosynthesis happening. This is, I think it's fascinating actually. This is equivalent to what photosynthesis is. So photosynthesis, we have a plant that have like a pigment in their leaves that absorbs the light from the sun, the visible light, as my colleague just presented, and turns this into energy, into chemical energy. So they were observing that some of those fungi, they were absorbing this radiation from the core, and they were turning this into chemical energy. In other words, they were eating radiation. So they were feeding on it. That was a source of energy as bread is for us right here. So that means that they need a pigment as well. And they found out that there's a pigment that's very common in nature. It's called melanin. That's the pigment that gives those fungi this dark color. That's why they're called black fungi, because they have so much of this melanin that they change colors. And this pigment was responsible for absorbing the radiation, and it actually was being used biochemically to generate this chemical energy that the fungus could use to feed. Melanin is not exactly unique from my slides. I'm not sure. Sorry, my slides were stuck. So melanin is not exclusive to fungi. All animals, as well as fungi, they have melanin. I'm showing here some animals that have excess melanin. This is actually, I'm showing then animals that have a condition, a genetic condition called ultramelanization or melanism, and it's basically the superproduction of melanin. And I'm showing them because of the dramatic effect, because few things are cooler than showing a black rooster like this. This is amazing. But all animals actually present melanin. We have melanin as well in our skin, and we actually use it as those fungi to protect us from radiation. So when you go to the beach and you are exposed to the sun, after a few days you start getting tanned, and that's actually your skin producing a lot of melanin. And the melanin is built as a response to the environment. In this case, the ultra exposure you are having to the ultraviolet light from the sun. And the melanin role is to protect you from this radiation. So instead of damaging your DNA, the UV will be absorbed by the melanin, and you're going to be protected this way. Some people don't have as much melanin, so when they go to the sun, instead they look like bell pepper, but you can't have everything, right? So in that way, melanin, it acts as some sort of sunscreen for us. Here we have a picture of a normal camera on the left and a UV camera or ultraviolet camera on the right, and it shows someone using sunscreen. There are some really cool videos on YouTube if you want to look for it, like showing how sunscreens actually work. There are two kinds, one that reflects the ultraviolet and some that absorb it like melanin does, which is the case here. So you see that this woman is spreading some sunscreen on her face and it becomes black. It's black because the sunscreen absorbing all the UV light that's being projected on her and it's not reflecting it back. So melanin does the same. Actually, if you look at her freckles, that's exactly melanin. So freckles in general are large deposits of melanin that we have on the skin, and that's why they look darker in the UV light. So in this case, the melanin is protecting her from the UV light the same way as the sunscreen is protecting her from the UV light. But I'm talking about fungi, not about sunscreen. And in fungi, melanin has a lot of roles, and it's very important for the metabolism. I'm showing here pictures of the fungus I work with, actually, and it's one of the space fungi that I'm going to mention later, and it's called Cryptococcus neoformans, and it has the ability to produce melanin when exposed to certain environmental stresses that can cause, for instance, exposition to radiation. For Cryptococcus, and as it is for other fungi, it's known that melanin protects from radiation, as I mentioned, and not only UV, but also all sorts of ionizing radiation. It also protects it from free radicals. So intense sources of radiation can actually generate free radicals in water. And these free radicals are very damaging to the DNA as radiation is itself. So these free radicals are a problem, and melanin, as well as other antioxidants that are produced by the fungus, are responsible for dealing with them and making them harmless, even with high amounts of radiation. And it's quite interesting because the free radicals is also a strategy used by something else that wants to kill fungi, our immune system. Our immune system uses oxides and other forms of free radicals to kill fungi. So our immune cells, when they try to kill fungi or fungal cells, they use free radicals, oxidative and nitrosative stresses. And that makes fungi that have melanin more resistant to the immune system. Actually, there are a lot of different researchers showing that melanized fungi are more resistant to immune cells, such as macrophages and others that are responsible for killing those fungi. Some studies, some really recent studies, have also been shown that melanin sucks the antifungal drugs, like it soaks in as a sponge and capture those antifungal drugs and make the fungi more resistant to them. So if it's a fungal cell that's causing disease, with melanin, you need high doses of drugs to actually kill it. So it's kind of a Swiss Army knife. Seriously, it does everything. It's good for everything for the fungus. It also protects against also some environmental conditions, such as temperatures or the presence of heavy metals that are known to actually kill fungal cells. I was talking about Chernobyl, but I'm going to jump now to another point in history. 1997, a couple of years after the Mir space station, the Russian space station that came before the International Space Station was finalized, and it was in operation until around 2001. So it was flying since 1986. So it was in orbit for 11 years. When in 1997, they started realizing that they were having way too many electrical problems. So they were having short circuits, and some panels were just dying or burning or even risking like a fire starting. And one astronaut at some point decided to look under the panels and started opening them up, and they found a large amount of cells of mold growing in the wires. So this is a picture of the time. So not great, but I think you can appreciate that those fungi, they are hygroscopic, so they capture water from the air. And so they were growing in those panels and then the wires under the panels, and they were capturing water from the air and making the whole environment a little bit more humid, and that was causing electrical problems. And so as soon as they started realizing that, they started having to replace those parts that were getting deteriorated. Later, they also start finding that some cells, some fungi growing in every single compartment in the space station, including the rings that are responsible for the ventilation of the windows to the outside. Here it's we have an image of actually from the International Space Station. This is not from the Mir. This is a way more recent picture, and it's an astronaut scraping a fungal. Some fungus growing, some mold growing on the outside of this window. So it was growing inside, started growing inside in between the titanium plates and eating on the rubber that's supposed to keep the air locked, the window closed from vacuum, and started growing on the outside, causing including a gas leak from the space station atmosphere. So that's a serious problem. You're having problems with the wires. You're risking having fire in your space station, and then you start having gas leaks, and then you have to replace modules way more frequently than people expected because you had this. Oh, I didn't move. Because they didn't. Sorry, they didn't. They were not prepared for that. And then they started wondering, like, where is this fungus coming from? And the fact is, fungi are everywhere. They are in our skin. They grow feeding on our dead skin. They are present in the atmosphere, the air we breathe, the food we eat. So everything we take there, the experiments we did in the space stations, we are bringing them along with us. Some research, research that was done in the International Space Station, where they tried collecting all the fungi they found there and comparing which species they were finding there with what we find here on Earth. They saw that the fungal species they were finding were exactly the same you would find in a bus stop or in a bus station or in a metro. So they are exactly the same fungi. We are carrying them with us everywhere we go. So wherever we are going, they are coming with us. But they were really intrigued about this whole thing of fungi growing outside the windows of the space station, including being exposed to the vacuum of space and to increasing amounts of radiation that are they would be exposed to when they are outside the space station without the protection of the structure of the space station. And so in 2012, they did one very interesting experiment in which they wanted to look at the viability of some of these fungi outside. So basically, they collected two different species. They are lichens. The lichen is some sort of symbiotic relationship between two or more species. They got some fungi that form this kind of symbiotic structures. And they are from Antarctica. They are from the Cryomyces species, as I mentioned here. And they put it in the Expose-E module of the International Space Station, exposed to the environment. So basically, it was exposed to vacuum, it was exposed to extreme cold temperatures, extreme temperature fluctuation, also all the solar radiation that's found outside, as well as cosmic rays and everything that you actually have in space. And they kept it there exposed for 1.5 years. So I'm not talking about a few days, it's for a long period of time. And they found out that after that, they recovered the fungus and they scrapped it, and they tried growing it again in like rich conditions in a nice medium when they were back in the lab on Earth. And they saw that a lot of the cells were still viable, even spending 1 year exposed to outer space. 4 years later, in 2016, they did a similar experiment, but this time they collected species of fungus that they found growing on the space station, not some random species from Antarctica that they knew already was super extreme. And they got those strains, those species of fungi that grow in the space station, and they started trying to grow them, not grow them, but like putting them in different surfaces. These different surfaces actually being the surfaces that are present in the space station, like different alloys, different materials that they can find in the space station. And they expose this to up to 5 months to the external conditions of outer space. And after that, they tried growing them to see if they were still viable for growth after that. And they were. So 4 different species were tested, and the 4 of them were still able to grow after spending 5 years pretty much exposed to the outer space. And then what can we actually learn from these guys besides being very badass? And what should we learn? The first thing is that they are a risk to the astronauts. Some of these fungi can cause diseases, and obviously they can cause structural damage to the space station. And if you think about a longer mission away from Earth, away from the orbit of the Earth, you have to take those things into consideration, that you're going to have degradation of some of the structures of the spaceship en route to Mars, for instance. Also, the risk to the ecosystems. And it's plural, because it's not just the Earth ecosystem or the space station ecosystem, but also the danger of bringing some strains of organisms like fungi that can grow, let's say, in Mars, and then they colonize that environment without any sort of restriction. Think about all the experiences we had on Earth in history, like when they tried bringing new species to Australia, and you will realize that we humans, we are really bad of trying to control how ecosystems work. So even Mars, that's an ecosystem that's pretty much untouched. As far as we know, there's no life there, and I don't think there is. But if we do bring some sort of life there that is actually able to grow, it would definitely take over the whole planet, and we don't know the consequences to that. And finally, those are really interesting qualities, some really interesting features that those fungi have that we could definitely be using in terms of technology for space exploration. So I'm going to take a quick look at those three items here. First of all, the risks to the astronauts. Some of these black fungi, some of these fungi that have the ability of melanization and becoming darker, they're actually, they can be a threat to the astronauts as well. So in general, fungi, they are not a pathogenic organism, meaning that they don't cause disease if you are a healthy human being with an immune system working as it should, but they can be opportunistic. That means that when your immune system is not working as it should, they can actually infect someone and cause disease. So I work with Cryptococcus neoformans, this really nice round yeasts that you can see here. And if we tested this audience, we would see that 80% of us has been exposed, and we probably are exposed to this fungus daily. We are aspirating those cells almost every day. But our immune system never let them become a serious disease. But this is one of the most important pathogens for AIDS patients, because one of the characteristics of the disease is actually having an immune system that's not fully operational, let's put it that way. And among those patients, this fungus can actually cause a lot of havoc and cause a lot of deaths. For instance, Cryptococcus causes around 200,000 deaths per year, mostly among AIDS patients. Another fungus that's found everywhere in the space station and in Chernobyl, actually, Aspergillus fumigatus. One of the most recent data that I found about it says 500,000 deaths per year, but I think this is a little bit exaggerated. These numbers are from 2010, and it's definitely less than that. But anyway, they do cause a lot of problems, and they can cause a lot of problems in patients that are immune suppressed. And one of the main characteristics, and one of the things that we observe among astronauts actually, due to being exposed to radiation, as well as the lack of gravity and the effects that it has on their physical shape, it has been observed that the immune system is actually impaired slightly. So these fungi could, in the long term, actually be a threat to these astronauts. The ecological aspect was tested. One of the experiments that was, I find it very interesting, is the Biorisk experiment that was performed in Russia between 2006 and 2009. And basically, they just took some different fungi. They did that with actually lots of different animals, insects, crustaceans, and different things. But I'm just talking about fungi. So they had the fungi in these plates, in this cloth, and they would be put in a cylinder like this, and this is placed in outside the International Space Station like that. And it was left there for up to 1.5 years. And then they analyzed the morphology of those fungi, and they observed that even though they were not growing while exposed to this space, when they came back to Earth, they had many different morphological differences to the fungi on Earth. So they had way more melanin they expected. They had bigger capsules, which is a structure that's important for preventing the immune system. So they had morphological differences, and they could have an impact on the ecosystems on Earth if they were to be released. So that was the first study suggesting that those that should be a quarantine on space ships or anything that actually goes to space after they come back. And finally, the technologies that we can learn from these guys, that we it has to do with, first of all, protection against extreme environments. If we are one day to colonize Mars, we need to find a way of protecting our colonies from radiation, for instance. They don't have a magnetosphere like we have on Earth that protects us from the radiation from space. And one way, actually, this is a picture from the NASA website, and it's one plan to make bricks, to make houses out of fungi. So those are bricks made of mycelia from fungal structures that are to be used as like building blocks to houses or buildings on Earth. This is a similar structure. Also, fungi are important resources, can be important resources as food, production of pharmaceuticals. The first antibiotic was produced by fungi. Also materials for construction and many other things. And those are very interesting for like colonizing Mars, for instance. And also they will be important in development of sustainable life support systems, both in a colony and also when they start having some environmental engineering whenever we start colonizing Mars. Maybe one day we'll be able to use those to actually terraform Mars. And with that, I will thank you for your attention. And I have this nice picture of fungus, of a mushroom that's called alien egg, because I thought it was proper. Thank you very much.
Thanks, Daniel. Fascinating talk, really interesting. We already have a lot of questions in the chat. So first question, is it a bigger concern to take fungi to outer space, or that we might bring new fungi back to Earth?
I think in a sense of bringing a new fungus back to Earth, in a sense that we are exposing something to radiation, and it will modify those fungi, and then you bring them back to Earth, and they can actually affect our ecosystem. I think both are a problem. That's what this last experiment I mentioned, the Biorisk one, talks about. They were afraid that exposure to radiation would modify them so much that when they come back, they would have some sort of advantage to the ones on Earth, and they could unbalance the environment here. And yeah, I think taking them out also is problematic. Like we might end up finding a fungus that can grow on Mars or something like that. We haven't yet, even though there are some reports that some damage done to the Mars rovers was actually caused by bacteria and fungus growth in the structures of said equipment. I don't know if that answers your question, but.
So there are a lot of other questions. Bacteria have been found in asteroids. Have fungi been found in asteroids too?
I'm not so sure about the bacteria on asteroids. I mean, that would be probably almost a proof of life outside Earth, right?
Yeah. Also, I'm not aware of these findings, but I'm
yeah,
not a space expert.
Unfortunately, I'm not a big expert on the topic of exobiology, but I would love to know about that. I don't think, so far, I've never heard about fungi and asteroids or anything like that.
So is there a set of genes or mutations that give these fungi this ability to live in radioactive regions?
Well, there are some genes like the melanin one or anything that upregulates or make the melanin production more efficient or more excessive, let's say. The more melanin, the more protected they're going to be. So I would say that those would be protective. And yeah, I'm not aware of anything else in this case specifically.
So Fritz Sedlacek wants to know, for these fungi in these harsh environments, do you observe a higher mutation rate across the entire genome or mainly on intergenic regions?
Well, I would expect, I don't know if anybody actually looked at the distribution of mutations like this, but I would expect to find them more homogeneously distributed across the genome, not necessarily intergenic. And the reason for that is that those cells, even though they are exposed to space and they are not dying, they are also in some sort of hibernation. So they're not fully active in terms of metabolism. So that's important because with the fully operational, if the metabolism like fully operational, they would have the whole repair system repairing all damage that they find on the DNA, and that's not the case. So you would be having like mutations happening everywhere at the same rate.
There's one question about opportunities for undergrad work on your projects. I think James Lively asked it, so maybe James, you can just contact Daniel directly, maybe via Twitter, or you're gonna find his email address at the WashU webpage. So just contact him. I'm sure he's happy to answer this question.
I believe my boss is going to be watching this, so I'll tell her and keep her. But again, we are not working here with space. We are working with fungus that's found among those space fungi that I mentioned, but we are looking something more medical application though.
So for the moment, I think we stick with the science questions then. So Marlene wants to know, have the fungi been able to be genetically manipulated to not be pathogenic? And if so, has their morphology and activity of the modified fungi changed upon space exposure?
Yeah, but that's pretty much what my project is about, is actually finding genetic modifications that make them less or especially more virulent. And among strains from different places from Earth, actually. Once we do find those, definitely, I think we can use this fungi to do almost anything. Like something I was going to mention, but I was running short on time because I talked too much. When I was a kid, I had an uncle that was diabetic, and he had to take insulin from monkey, horse, cow. Like they had insulin from animals at the time, and that's what he had to take. And people would have like side effects to those, and they would have to keep changing from one to the other, and it was always terrible until like afterwards in the 90s they found out how to make fungi and bacteria produce human insulin for us. So in the future, we're definitely going to be using those fungi to actually do what we need to do.
So Francesca wants to know if you have any thoughts about the Star Trek Discovery traveling through space using a mycelium network.
A what?
A mycelium network? See, it's much harder for me to ask these questions when they are so biology related.
Yeah, mycelium, like being the structure of mushrooms. I didn't watch the series, so I don't know much about it, but what is that, like a bioship concept or?
Oh, I don't know that either. Maybe Francesca has to send you an email as well.
Yeah, please do.
We have another more science-related question. Is there a difference in mutation evolution rate between fungi on Earth and in space?
It's a hard question because, again, when they are in space, they're metabolically not fully active. So they are being exposed to mutations, and it can happen that some of those cells, when they come back to Earth, make them have a higher fitness to survive in the Earth environment. But it's kind of a gamble. It's not like a Spider-Man thing that you're actually, it's the Hulk, that you are exposed to gamma radiation and then you get superpowers. It's more like 99.99% of the mutations are actually going to be very bad. And in this case, I'm not sure if those mutations are helping the fungus in space to be selected there. They're just causing the mutations, and when they come back to Earth, that's when we're going to find out if that was good or bad.
Great. Yeah, I think we're already a little bit over time, but it's not a problem online, of course. I think if you have time, please fill out our survey. I've posted it just now again on the chat. Daniel, thank you so much again for your talk, and also to our first speaker, Johanna, thank you so much. And please check out our Twitter. Be sure to not miss our next events. We don't have a date set yet, but we're going to have more events in the future. And thanks for joining us today.